Okay, and I'm going to go off my camera. So thank you so much, Taylor, for presenting. Take your time, and then we'll field questions at the end.
Okay, can everyone see my PowerPoint right now?
Yep, it's good.
Perfect. Yeah, and I'm glad it's being recorded because I'm wanting to go back and see some of the recorded talks as I've had some conflicting meetings on previous months. So unfortunately, this is my first time to this meeting. But yeah, as Adam introduced me, I work at the National Genomic Center for Wildlife and Fish Conservation, so that's nested within the research branch of the U.S. Forest Service. And today I'm going to talk about high throughput qPCR for targeted and parallel analysis of environmental samples, specifically focusing on invasive species. But first, I want to talk about what I think are two important attributes of invasive species, specifically when we think about eDNA sampling. The first is that they're diverse. When we talk about aquatic invaders, we're talking about everything from plants to mollusks to fishes to mammals. And then when we're talking about eDNA sampling, we're often interested in rare individuals. We want to pick up organisms at low abundances before populations become established and start causing economic and ecological damage. If we look at our kind of molecular toolbox for eDNA sample analysis, we see that we have some tools that are a better fit for each of these components of aquatic invasive species. So on one side, for diverse threats, non-targeted analyses, things like metabarcoding, which can cover tens to hundreds of taxa within a single analysis, are really powerful. They can be really cost effective and allow us to look for whole suites, kind of taxonomic groups with a single analysis. On the other side, when we're talking about rare individuals, some of our targeted analysis tools are probably the best fit. So things like digital droplet PCR and qPCR, because these can achieve single DNA copy sensitivity. They're really specific. We can optimize these analyses for the specific taxon that we're looking for. What is a potential challenge is that these two tools, although they're really good for each side of this, they're not kind of universal solutions, or at least they're not optimal universal solutions. So non-targeted analyses are great at covering diverse threats. They're not the most sensitive approach for detecting rare individuals. Conversely, targeted analyses like qPCR are great at picking up rare individuals, but they're not going to be cost effective as we start to get into tens to dozens to hundreds of taxa. It's really just a one to a couple of species type tool. Optimal solution would allow us to cover diverse threats with the same kind of sensitivity that we have for targeted analysis for detecting rare individuals. And so in this talk, I'm basically going to talk about how high throughput qPCR might in some cases start to approximate this optimal solution. So for the rest of the talk, I'm going to give you a little outline that I'm going to come back to help us keep things straight. First, I'm going to do a quick recap on how qPCR works, so we're all on the same page, and that'll help us to talk about how high throughput qPCR works. And the big keywords here are miniaturization and parallelization of qPCR. Then we're going to talk about how high throughput qPCR performs, benchmarking against single species qPCR. Then we're going to talk about two different projects where we're applying high throughput qPCR to AIS, mostly an update on these projects and not a whole lot of final results to share at this point. And then finish with noting some of the uncertainties that remain. Maybe this can jumpstart some discussion if folks want to get into the nuts and bolts a little bit, or think about what some of the considerations might be as we apply these to management type situations, and a note on where we think we're going next with these tools. So first, let's do a quick recap on qPCR. This is a illustration of how PCR works, polymerase chain reaction. The black parallel lines are indicating double-stranded DNA. The red arrows are single-stranded DNA called primers. Over multiple cycles of heating and cooling, these primers anneal, extend, and make millions of copies of our DNA, the black lines again. And detecting this amplified DNA, these PCR products, is how we know that we have the DNA that we're interested in present in a sample. qPCR typically uses a hydrolysis probe, sometimes referred to as a TACMAN probe, which is another single-stranded piece of DNA, an oligonucleotide that binds downstream of the forward primer. And as long as this oligo is intact, there's no release of fluorescence. Specifically, this probe needs to bind with basically a perfect match to the target DNA in order to anneal tightly. So for example, this is supposed to be an illustration of an assay for lake trout. You can see that with the colored bars here on the probe that we have perfect complement, a perfect match to the target template. And when we have that perfect match, when the forward primer extends, it hydrolyzes this probe, it separates the reporter and the quencher, and there's a release of fluorescence. And that's how we know that our target DNA is present. Conversely, if we have some non-target species, for example, bull trout, this probe might sit down on the template, but because it's not bound to it with perfect affinity, it's going to just be displaced by forward primer extension, and we're not going to have a release of fluorescence. The result of this is that as we proceed with cycles of PCR, we have this characteristic sigmoidal curve of fluorescence when there's target DNA. So those are the gray-blue lines. But if there's just non-target DNA present, we're not going to get an increase in fluorescence. And I think it's important to note that, as I kind of alluded to earlier, no other tool is as sensitive or specific as qPCR. I'm not going to get into the nuts and bolts of why that is, but there's been multiple papers, I've linked to a couple here, that show that when you take the same environmental samples and you run them on qPCR versus on metabarcoding, that you have better detection for rare species using qPCR. The little asterisk here is just noting that digital PCR, digital on a microfluidic chip or digital droplet PCR uses the same chemistry, and it's going to be really similar to qPCR in terms of sensitivity and specificity. So now we've done a little recap of how qPCR works, and that's going to help us understand how high throughput qPCR works. High throughput qPCR basically just works by reducing our qPCR reaction volumes to less than 0.1% of a conventional analysis. So here again, I'm going to use kind of a cartoon to illustrate what I mean by this. Here, the blue rectangle is supposed to represent a conventional qPCR 96 well plate. So we can do 96 reactions on one plate of qPCR. On a biochip, a high throughput biochip shown by the orange square here, those 96 reactions just take up a small proportion of about 5,000 reactions that we can run in parallel. So that's going to be this little blue inset in the bottom left corner of the chip. We have an actual picture of these in the top right of this slide. So in the background, you can see there's a plastic PCR plate. And I think this is a 384 well plate, but it's the same size as a standard 96 well plate. And then in the foreground is a aluminum biochip for the high throughput analysis, and that has 5,000 reaction wells. And here there's just a robot that's loading samples and assays from that 384 well plate onto that 5,000 inlet biochip. So the reason that we go to a high throughput platform is that we save time and money. And the way that we do is through two mechanisms, that miniaturization and parallelization. So miniaturization refers to the fact that because each well is tiny, we don't use as many molecular reagents per reaction. So right there, we save money, expensive molecular reagents. Parallelization refers to the fact that because these wells are so tiny, we can put 5,000 of them on a chip. And so we're able to do many analyses all at the same time. And this saves a lot on technician time, rather than needing to do iterative analyses for samples, or to do many analyses on one sample and then iterate that across samples. So through miniaturization and parallelization is where we see cost savings on a per taxon per sample basis compared to qPCR. And I'll talk a little bit more about costs here in a minute. All right, so we covered qPCR, how high throughput qPCR is just taking qPCR and downsizing it through miniaturization and parallelization. Now we're going to look at some examples of how high throughput qPCR performs, benchmarked again against single species qPCR. So this is a published example that we did about 2 years ago. Here we took 524 samples from the range-wide bull trout eDNA project that had already been collected from across western Montana. That's what this map is showing here, are the sites per watershed indicated with color, and then had been analyzed on a single species qPCR platform. And then we analyzed them on high throughput chips that included bull trout markers. We saw that concordance between qPCR and high throughput qPCR was 90 to 99%, breaking this up chip by chip. And this level of concordance is similar to what we see if we take one bull trout eDNA sample and we run it on qPCR, and then we run that again on a second plate of qPCR. There's some level of variance run to run, just because copy numbers tend to be low, often less than 10 copies per reaction or less than 3 copies per microliter in the extract. And so there's some stochasticity there. We then specifically zoomed in on 2 watersheds here. That's indicated here with the blue rectangle, Rattlesnake Creek, to look at concordance across multiple different markers. So here we have panels showing a map with 48 sampling locations in the top left for bull trout, then brook trout, rainbow trout in the bottom left, and brown trout. We have circles that are split in half. Solid blue means that both qPCR and high throughput qPCR detected the taxon. Yellow means we didn't detect them. And then there are a couple of cases where we have discordance between the methods. And these are indicated half and half with a red asterisk drawn to the top so we can see them. We can see that across these 4 different assays on these samples, we had concordance that ranged from 95 to 100%. So again, that's similar to what we see when we run a sample iteratively on single species qPCR. We also had known composition samples where we knew how much DNA was present because we spiked it in. And we saw that we had 100% amplification rate on the high throughput platform when there's at least 3 copies per microliter. And 3 copies per microliter is just the bottom dilution that we actually tested. I will note that we found that there was one assay that had poor performance when we put it on the high throughput qPCR platform. And that was an assay for West Slope cutthroat trout that I don't have results for here. That was kind of a flattened amplification curve that made calling positives potentially ambiguous. And so we didn't include those analyses here. So now I'm going to look at another example. This is a chip that we're working on right now. So I'll emphasize that these are preliminary data as we're building up these confusion matrices and going through the details of this. This is work that I should note is being led by Joanna Elmore, working with Kelly Kareem, Mike Young, and with Trout Unlimited in Washington. Here we have a chip that's composed of 9 Pacific salmonids as well as Pacific lamprey. Those are shown on the x-axis. There's a picture of each taxon, and then above each there's a number which indicates the number of environmental samples that are used in our qPCR to high throughput qPCR comparison. On the y-axis is our estimate of concordance. The individual dots are our point estimate for the concordance, and then we have confidence intervals based off of the lines are 95% confident intervals based off of bootstrap resampling. So what you'll see is that on average across all of these assays, our concordance is good. It's around 93%. We do potentially have an issue here with the coho salmon assay, and that's something that we're looking at right now. If this has to do with lower sensitivity on the high throughput platform, or if this has to do something with the specific assay and what its limit of detection is across platforms and the specific samples. So we're going to be about doubling the sample size here before we make a final assessment on that assay. All right, so in general, I think we've seen that high throughput qPCR chips, and these are just 2 examples of some that we've run recently in the lab, show similar performance to single species qPCR. The caveat there is cases like the West Slope cutthroat trout and the coho salmon, where there may potentially be assays that need some additional attention in order to get good performance on the high throughput qPCR platform. And I'll talk about this a little bit more, but I think this is a place where we need a little bit larger sample size to help us identify those assays that will need some optimization before we can port them over onto a high throughput chip. All right, and then also this should be taken as very preliminary, but let's think about performance in terms of cost. Here's a table where we lay out 5 possible chip configurations with the current platform that we've been using, where we have the number of assays and the number of samples that would be run per chip, an estimate of what the cost per sample would work out to be, and what the cost per taxon per sample would be on that last column. For reference, at the National Genomic Center, we typically need $85 per sample for qPCR, and then $35 for each additional taxon after we've done that first sample extraction, archival, and test for PCR inhibitors. So if you look at cost, you see that the high throughput qPCR platform is going to have a per taxon cost that's on the order of 1/10 to 1/5 the cost single species qPCR. It's definitely going to be more expensive than metabarcoding on a per taxon basis. So depending on how you benchmark it is going to dictate how you think about it as succeeding in cost. Again, we were thinking of this as being benchmarked against qPCR because that's what we typically lean into for AIS surveillance because we put a high premium on that detection probability when species are rare. And this is just some text reminding me to say basically what I said, other than that kind of a threshold between qPCR and high throughput qPCR is probably around 3 taxa. If it's 2 or fewer taxa, you should just use conventional qPCR. If it's 3 or more taxa that you're interested in, then these high throughput chips are going to start looking cost effective. All right, so we talked about how high throughput qPCR works, and now we're going to talk about some examples where we're applying this technology specifically to aquatic invasive species. First is a Department of Defense funded project called Validation and Implementation of an Aquatic Invasive Species Biochip. The idea here is that we are to validate about 50 aquatic invasive PCR assays and then package them together onto a high throughput chip so that partners from across the lower 48 United States can run samples on a single protocol that will screen for a wide range of aquatic invasive species. An important part of this project is on technology transfer. So some of the products that we're anticipating from this are not just the assays, but a published peer-reviewed protocol that someone can use directly from the bench to run these biochips in their own laboratory, and then having a proficiency testing program so that we can allow other laboratories to try the protocol and demonstrate that they're able to follow the protocol and reproduce analyses on samples that they're blind to but that are known composition submitted to them, which is a model that we've seen, for example, with the UK greater crested newt surveillance work. That's a native species, but in their monitoring work. So excited about this project. Right now, we're early in this project. We're validating assays. We're going as fast as we can while doing a thorough job of characterizing existing published assays and developing some new assays for application across the United States. And the planned completion for this project is sometime in 2024. So that's when we'll be thinking about having that protocol out and initiating our proficiency testing plan. The other project where we're applying this technology is for the U.S. Forest Service Regional Aquatic Invasive Species Monitoring Strategy in the Pacific Northwest region. This is being led by Jim Capurso on the field and the management strategy side, and John Cronenberger in our lab on the technical development side. Right now, this is a 10-species chip with some taxa that are of particular conservation concern. Right now, we're analyzing samples that were collected in 2021 and comparing our concordance between the high throughput platform with single species qPCR and coming up with some best practices for communicating those results to our management partners, which particularly to the individual forests where these samples are coming from. But the planned goal for this project is to expand the chip to cover about 30 taxa that are a priority in Region 6 for the U.S. Forest Service, running about 100 sites annually on this chip as part of the overall AIS monitoring strategy. All right, and finally, I'm just going to note some uncertainties that remain about this technology, and happy to talk in more detail during the question period about what we're doing to address those and where we see that we're going next. So this first one I already mentioned earlier, which is, can we uniformly use existing qPCR assays right off the shelf? If we have a well-validated qPCR assay, can we just put it on a biochip? It looks like the answer off of the limited data we have right now is 90% of the time at least, yes. But there seem to be some cases where we have qPCR assays that don't perform as well when we put them on the biochip. And so we need to explore further what characteristics of those assays make them not perform as well, and can we predict those in advance so that we can improve our design, or we can at least flag those assays for potential redesign before we incorporate them into chips. Another question is, how do we monitor for inhibition in high throughput qPCR? This is something we have some data on now. In our most recent iterations of these chips, we now include an internal positive control to monitor inhibition in the same way as we do for single species qPCR. And that looks, just had a meeting this morning before I put this slide, or after I put these slides together, where that looks really good with the protocol that we have right now. Another question is, how accurate is high throughput qPCR quantification? Here I focused on recreating detection, non-detection data as we get it on single species qPCR, but for some questions we might be interested in eDNA concentration. Here again, we have some preliminary data where we've done standard curves that are multiplex across all the taxa on the chip, but I think we need some more data with a larger or kind of range of values to assess how consistently we get good detection results compared to single species qPCR. Quantification results. And then finally is kind of on the application side, we're still figuring out where this fits in with our toolbox. When do we use things like qPCR, digital droplet PCR versus high throughput qPCR or metabarcoding? We have started to flesh this out a little bit, what we think the landscape might look like as far as using these tools. On one end is that 1 to maybe 3 taxa range. If that's all that you're interested in, shown here on this top bar, then really single species qPCR is going to be our most sensitive and cost-effective tool. On the other side of this, if we're talking about hundreds of taxa, we really need to use a non-targeted approach. High throughput qPCR isn't going to cover those taxa well. It's in this middle ground where we're talking about a handful to several dozen taxa where we might decide between using high throughput qPCR and a non-targeted approach such as metabarcoding. First question is our taxa list defined? If our question is something like, how many species of fishes are in this river, then high throughput qPCR won't be able to address that. You need to go with a non-targeted approach because you have to define your species list. The next question is, how critical is rare species detection? I am arguing that for aquatic invasive species, this is really important. So if you can define your taxa list, there's a benefit potentially to going with high throughput qPCR versus a non-targeted approach. And then the final consideration is, do you have qPCR assays already existing that you can port onto the high throughput platform? It's a lot more R&D cost if you need to design new assays before you can assemble your chip. Then finally, where are we going next? The Aquatic eDNA Atlas project, if you're not already familiar with it, is a public geospatial database with about 22,000 eDNA sampling records from across the United States. There's a hyperlink here underneath that text, but I can also send that link to folks who are interested and haven't worked with this system before. These high throughput chips, including replication and controls, generate about 800 data points per chip. And we've been running chips over this last year and see some more doing in this next year. So we're looking at probably about 2023, incorporating data from these high throughput chips into the Aquatic eDNA Atlas. So that's going to be a pretty decent sized data dump on top of this existing database. So that's what I'm kind of looking forward to in the intermediate term. And with that, I'm happy to take any questions, have some discussion time. I just have a block of text here recognizing that there are multiple funders associated with this work, as well as really critical partnerships, especially underlying the eDNA Atlas database with federal, state, tribal, non-governmental, and industry partners. Yeah, thank you.